Dr. Hsiao is a postdoc at National Taiwan University, where he works on the systematics of beetles. He just received a doctoral degree from the Australia University, and he's going to present on systematics, diversity, and evolution of the Australian cycad-pollinating weevils. Hello, everyone. Can you all hear me? Okay. Thank you. My name is Hsiao Yun. I'm currently a postdoctoral fellow at the Institute of Ecology and Evolutionary Biology at National Taiwan University. My topic today is on the research I conducted in Australia in my gaining my master's degrees on the Australian cycads pollinating weevils. Cycads are palm-like gymnosperms. They originated in the Permian and were widespread and diversified during the Mesozoic. They were distributed in tropical and subtropical areas. And unlike other gymnosperms, cycads mainly rely on beetles to spread their pollen rather than wind. The Australian cycads species and subspecies, there are 85 named species and subspecies in 4 genera, and among them, other than Sayas that also were seen in Asia, the rest, Macrozamia, Lepizamia, and Bohemia are unique to Australia. And for Australian cycad pollinators, other than the weevils, the beetles that we're going to talk about, there are also thrips. And thrips beetles, and they are the pollinators for 12 species of Macrozamia, and 6 Macrozamia species are exclusively pollinated by thrips. And there are beetles and weevils, and other species are only participating in a smaller portion of pollination. So we will focus on weevils today. Weevils are a type of beetle that has a long mouth. That's the most diverse group of beetles, with more than 60,000 described species, and scientists estimate that there are more than 200,000 species. So there are a lot still unknown. And there are 4 usually associated with cycads. The trains group. So the first, Demucerus and Ceraton, these 2 trains, they are stem-eating weevils. So they're more like pests for cycads. And the other 2, myrtle trains and trains, they feed on the cones of the cycads, and many species are considered to participate in pollination. And how? When we cite from Macrozamia and Communius, we look at the relationship between the 2 species. So every year during the reproduction period of cycads, so during the day, when the volatile organic compounds released by the cycads are low, a large number of male weevils would be on the cycads, and they would feed on the pollens and on the cones. And during the night, the level of concentration of volatile organic compounds would be the highest, and the weevils would not be able to bear the VOCs, so they would leave. And as the level of VOC drops again, all the weevils would fly back, and a lot of weevils are carrying pollen along the way. And the cones, the pollens that the male cones with pollens and the female cones that do not have pollens can be pollinated because of the movement of the weevils. And the size of the weevils is very small, and we do not know the number of specific species yet. And the nanoplastic group is very similar to the species, and these are the research objectives. First is reconstructing lineage. We want to see how the co-associative evolution, co-evolution only occurred once, and we want to know the number of species, and we want to see if the cycad weevils are having a high level of host specificity, just like fig bee. Next, we want to know the time of origin of such evolution to see if the pollinators are of the same as old as the plants. And we want to know whether the cycad weevil pollination relationship would drive the synergistic development of two species. So the first part need to clarify the parental relationship or kinship. So we use a large number of museum specimens. We sampled all the known cycad weevil species, and we tried to select samples from different origins and different host plants. And in terms of sequencing, we used next generation low coverage sequencing. And Dr. Liu said that there was in her study the coverage was 80%. For my research, it's only 1% for each species. So we try to capture the lower side. And through bioinformatics, we try to capture the data we wanted, and we obtained 120 samples, and we downloaded all the genomes that are already known in our data sets, and we use IQ tree to find and the variations into IQ tree, and we want to see check the stability of the tree's form, and we sample the database, we adjust the sample sets, we adjust all the different variables to see if our tree form is consistent, and in the end it was. And we found the subspecies location, and we found that the Australian cycad weevils are not monophyletic. So for the weevils that are co-related, or they're co-evolving with the cycads that encountered in more than one evolutions. And we found that the Literias from Southeast Asia associated with pendants, that is quite similar to how trains are associated with country, and we found that the weevils pollinating for Australian cycads, they look the same or very similar to Literias in Southeast Asia, but they are not actually related. So in terms of form, that would be a convergent evolution rather than kinship. So these are the basic taxonomy. We reviewed all the samples from collection institutes in Australia, and we also we took field samples. So we can see that there's lots of difference in terms of their morphological studies. And we used different methods to examine delimitation. And in conclusion, we found 19 species that can be recognizable, that are also supported by morphological studies and molecular evidence, including 8 completely new species. And after knowing the number of species, we want to see if they are host-specific in pollination. So we mapped the hosts information, cross-referenced them, and we learned that most cycad weevils are not host-specific. They can pollinate with multiple species of cycads and even across 2 different genera. They seem to be able to utilize various cycad species within their region. So they are sympatric. So the different lineages, they can be sympatric or partially sympatric, but for the same species, it would be allopatric. Next, the evolution biology. We look at the existing fossil records, and we found the supported fossil records, and we dated time, and we used reliable fossils for correlation, and then we examined the data to see, and our results were quite consistent. The origin time of the cycad weevils are of the Miocene and Oligocene. So compared to the cycads that are ancient, all the weevils are relatively young. However, when we map or compare the time of diversification of cycads and weevils, and we found a synchronous diversification. So we want to find whether the coevolution would take place for cycads and their dedicated or obligate pollinators. And we found that there's synchronous diversification. So we want to see if that's a result of this pollination relationship. So we conducted a co-speciation analysis. We want to see if there is a large-scale co-evolution event. We used the ID tree that's published last year, and we focused on the lineage that has the highest diversity and that has the most research. And because the taxonomy of cycads are pretty tricky, so the variation would affect our research. So we would just combine all the similar species into one in the definition to see if it affects our results. And the result is that none of the analysis we did support the facts that co-speciation events would co-evolution took place between cycads and weevils. And even though there is a synchronous time in diversification, our research showed that our hypothesis was not supported or verified. There is no co-speciation took place. There will be different possibilities. The first, in co-evolution, there would be very consistent change or correspondence in function. For example, for big comets, orchid has a long spur, and the long beak hawk moth has a long beak, and that's corresponding. And another evidence is that for the fig bee doesn't have unlike the fig bee, the cycad weevils do not have a specific host species. So we determined that the geographic distribution may be the largest contributing factor to the synchronous diversification. And that may be more relevant to the the biological diversification events happening in late Oligocene common to Australian species. So in summary, first, Australian cycad weevils are not monophyletic group, and the associative relationship with cycads has evolved in many times in Australian weevils. And the cycad weevils are not specific in terms of host plants. In sequencing, we found that the cycad weevils originated from the late Oligocene to the middle Miocene, then are much younger than their cycad hosts. But the time of this diversification events of cycads and weevils are synchronous. However, after our co-evolution analysis, we found that geographical distribution may still be the main factor deriving the diversification of the both species. And the synchrony of speciation time between cycads and weevils may be similar to the large scale of biological diversification in Australian species, including snails and other species, that in late Oligocene. So this is my presentation. Thank you very much. Thank you for listening, and I too thank SSE for the opportunity. Thank you.
谢谢肖博士带来了精彩的报告。
Thank you for the wonderful presentation. I think in the chat box, Professor Zhang Kai actually asked a question. Zhang Kai, do you want to ask the question directly? Okay, I just want to say that when you are doing the presentations, in the background, you mentioned that the weevils that would actually release something called EUC that can repel weevils. So for this EUC, is it really specific or it's both broad spectrum? Does it have to do with the evolution of these species? Like you said, the co-speciation, does it have any relationship? Yes, because you mentioned that it's not matching. However, in terms of the smell, the odor, is it matching? That's what I want to know. First of all, in the weevils, in Australia and with the volatile odor occurrence, the study we only have the VOC actually by the cycad, and so there are actually 4 ingredients, and these are artificially generated since it's really strong, and that means that they can actually repel the weevils. Is this specific for a single species? Well, actually in Australia, before my study, they only have one type of weevils. They didn't separate it into different species. So it's hard to know if there's any specific relationship. But I think my analysis and my research already said that there's diversity in this weevil. Then that means that in the future, maybe we can, find more relationship between its relationship with volatile VOC, and we will see if that relationship is specific or not. Thank you. Hello. Can I also ask one question? Yes. I think this is a very interesting work, but I want to know that when you're studying this weevil, you said that this is actually a host. The host would be the cycad. So does the powder of the cycad also needs to rely on it or not? So all these, yes, they are actually specific to they will not go to other. So it is very specific to cycad. So does cycad actually need the weevils to do the pollination? Well, in terms of the research for the weevils, the relation between weevils and cycad, yes, many species actually rely only on the weevils to do the pollination because in the past, the studies shows that they try to separate these species from doing the pollination. But once that separation is actually included, then the pollination would start again, so the pollination would be higher. So if we control these isolations and non-isolation, if it's not isolated, the pollination rate would be much higher. So that's why it's concluding that yes, these plants do need weevils to help doing the pollination. However, there are different cycads in Australia, and all the research show that actually wind can also have a certain influence in helping the pollination. So it depends on which plants are you talking about. So this means that weevils increase the pollination rate of that plant. So that's quite interesting. But you're also saying that the research shows that your research doesn't see, the speciation, and there's no co-speciation between cycad and the weevils. Yes. Why do you think that is? Well, the reason behind what we do see that there's actually one type of weevil that can actually help two different types of plant cycads to actually do a pollination. So I think that it's more about, if whenever there is a fruit there, then they will go there and to do the pollination. But it's not like it seems like at least the weevils can actually fly anyway. So in terms of the luck relationship, I think it's not that luck that not specific because the weevils may still go to different places. I mean, the pollination may be still in the initial evolution stage for pollination specific. Okay, thank you. Thank you for that response. Thank you for that wonderful discussion. Dr. Hsiao, just a reminder that in the Q&A session, there's actually one question, so you can also type in the question directly.